So we've met the idea already of the central dogma theory in biology, and that is that there is coding regions within the DNA, and the coding regions within the DNA are transcribed into RNA, and that RNA informational element is then allowed to leave the nucleus, if we're talking about a prokaryotic cell, enter the ribosome either at the rough endoplasmic reticulum or free ribosomes, and that code is translated into a polypeptide. And remember that polypeptides we need to separate as distinct from proteins. But again, you can read this detail for yourself, which is what we've talked about already. What I want you to think about is to relate this to 7.1. And remember in 7.1, we talked about the elements of DNA which are non-coding and coding. The non-coding regions were once upon a time thought to be junk DNA. Junk is a misnomer because actually those particular elements of DNA are very, very important for a variety of functions, including getting involved in the process of transcription. So remember, we looked at transcription last time. We looked at this idea that RNA polymerase is the enzyme that both unzips the DNA. It uses one of the DNA strands as a template, and using complementary base pairs, it brings into position the ribose nucleotides to produce this messenger RNA strand. Remember also that the direction of transcription is in the 5' to 3' direction, and what we're talking about is the direction in which the new messenger RNA strand is produced, not the coding strand. We talked about transcription then of coding sequences. What we'll jump to in 7.2 is looking at the non-coding region, the promoter region, and the terminator sequences. So transcription is going to require the binding of RNA polymerase, and RNA polymerase to bind needs a kind of indicator of where and what to do, and this is where this promoter region comes into play. The promoter region is a section of non-coding DNA, but it is essential for transcription to take place. In addition, when RNA polymerase moves along the DNA gene, then it needs to know when to stop. So there is a terminator sequence, which is usually also non-coding, which terminates, as the name suggests, the process of transcription. So these two elements we come back to in 7.2. And what I want you to do is just make sure that you're familiar with these in terms of the work that we have done for 7.1. So transcription of the coding sequence. What we also look at in 7.2 is this idea of post-transcriptional modification, and in particular the idea of splicing and adding a poly A tail and a 5' cap. So what we do when we do transcription is we produce what is called pre-messenger RNA. And when we come back to 7.2, we look at how that messenger RNA is later modified within a eukaryote. But for now, let's just put that into the back of our minds. And then we do the process of translation, which is what we're going to focus on here today. So just to bring you up to speed and to remind you that RNA polymerase will only work in the direction of 5' to 3' in terms of synthesizing a messenger RNA strand. So this red here is coding for the direction in which this is being produced. Remember that RNA polymerase is both responsible for breaking the hydrogen bonds that exist between the DNA strands and also responsible for bringing in the RNA nucleotides or the nucleoside triphosphates more accurately into position, and we're using complementary base pairing. Remember also that uracil replaces thymine in terms of the messenger RNA. This is a template strand. This is also called the non-coding strand. This strand at the top here will have the same base sequence, albeit thymine being replaced with uracil, as this messenger RNA strand. So this top strand here is referred to as the coding strand. This bottom one is the non-coding strand, which is also the template strand. Just to remind ourselves of what types of RNA there are, we've got messenger RNA, which is this single-stranded informational RNA. And messenger RNA is made up of a series of so-called codons. And codons are 3 bases which are adjacent to one another. And one codon, or a triplet of 3 bases, is the code for one amino acid. And that becomes important when we look at this process of translation. Now, the codon is the same across all species. So the DNA code, and specifically the way in which messenger RNA is translated, is universal across all species. We've also got this molecule called transfer RNA, and transfer RNA is the molecule that is responsible for bringing the amino acid into the correct position at the ribosome. And transfer RNA is also single-stranded, but is folded up into a particular shape, which we'll look at in just a second. Then we've got ribosomal RNA, and ribosomal RNA is, along with protein, is making up the actual ribosome. And this has got some kind of catabolic function, allowing you to produce a peptide bond when you're doing translation. So just to remind us of transfer RNA, so you've got this so-called clover leaf-like structure, which is a three-dimensional molecule, and the loops in particular are very important because that allows this transfer RNA to specifically bind to a particular enzyme, and that enzyme also has an active site which will fit a specific amino acid. So a specific amino acid will be attached at this 3' end of the molecule. In order for that attachment to take place, you need to use ATP, so the process is an active process, and the amino acid is attached to this 3' end, which is very, very specific to this particular transfer RNA molecule. The important thing to note is this part at the bottom here on loop 2, and loop 2 contains the so-called anticodon, and the anticodon is also a triplet, and the anticodon needs to be brought into position with the amino acid which is appropriate to it, to the codon on which you've got complementary base pairs. So you've got complementary base pairing between the anticodon and the codon at the site of the ribosome. If we look at this molecule, this diagram rather here, we can see that we've got the ribosomal units. We've actually got a small unit and a large unit, and we've got within there 3 particular sites. Now, these sites don't actually exist. They are models which allow us to explain what's going on. And what you can see is that this is the messenger RNA molecule. The messenger RNA molecule is held between the small and the large subunit, and that messenger RNA molecule is pulled along until it hits the so-called start codon. And the start codon, again, it's a triplet of bases. This is it here. It's always AUG, and the start codon is universal again across all species. And AUG will allow a particular messenger RNA, which is denoted by this here, and the complementary base pairing will be UAC, and UAC on this anticodon will always mean that it carries methionine, or formal methionine is the same thing. So that is being brought into position at the so-called P site on the ribosome. There are 2 other sites, the E site and the A site, which will become clearer as we work through this slide. So just to remind you that we've got 3 different types of RNA. We've got the messenger RNA, this single-stranded protein here. We've got transfer RNA, which is also single-stranded but folded into a particular shape with a particular anticodon and a very, very specific amino acid which is bonded to it. We've also got ribosomal RNA, which is making up the ribosome itself. So the genetic code is said to be universal. So the codon is going to be the code that is going to be the same whether you are a Drosophila, whether you're a fungi, whether you're a prokaryote even, or whether you're a human. So if you've got AUG as a codon, it will always bring into position the amino acid formal methionine. The code is also said to be non-overlapping. So if we look back at this diagram, it means that the codons are read as a triplet here, and the next triplet is going to be these 3 here. So this triplet is next, in which case we're going to shift down a whole triplet. So what we don't do is say AUG and then look next at UG and, for example, C here. So we don't overlap this. It's 3, and then we slip along, and it's the next 3, and it's the next 3 that are read. So it's a non-overlapping sequence. The sequence is also said to be degenerative because there are more than one transfer RNA molecule sometimes which can code for the same, or which can carry rather the same amino acid. Excuse me. So there's more than one codon that can code for the same amino acids in some cases, which is what degenerative means. So this is a table which allows us to look at the different codons which code for a particular amino acid. Now, take care with information that's given to you, because sometimes this could be a table which is actually specifically talking about the DNA code, but in this case, I know it's messenger RNA because of the presence of U. And the way in which we read this code is we look at the first letter to begin with, and then we look at the second, and eventually we find the third. So let's consider AUG. So the first codon is A, the second codon is U, so we go across, and then we come down here. So we've got here, and the third codon is G. Here we've got methionine, which is the initiation always of protein synthesis. So AUG is the codon, and that codon always is going to code for formal methionine. Going back to this idea of degeneracy, you can see that this codon here, CUU, CUC, CUA, and CUG, is always coding for this amino acid, which is leucine. So in some cases, you've got many, many different types of codon which will correspond to the same amino acid. But that is the reason why the code is said to be generative, and it's not necessarily true of all codons. So again, go back into the Google Classroom and read this at your leisure. But what we need to appreciate for the syllabus is that codons, which are 3 bases on the messenger RNA, correspond to 1 amino acid in a polypeptide. In an exam, they could give you some data like this. It could show you the codon sequence. It might show you the anticodon sequence, and it actually could show you the DNA sequence. So you need to go back and forth and make sure that you're clear in your mind that the DNA on the coding strand is going to be the same, albeit with uracil instead of thymine for messenger RNA. Again, you can interpret this code. So it's degenerative. Also, there are some regions which are, some codons rather, which are saying it's the start codon. It's always AUG across all species. So we've got this universality. And also to bring into your attention that there is a stop codon, and the stop codon does not correspond to an amino acid. So when the messenger RNA is read and it hits a stop codon, then that will stop synthesizing your polypeptide. We'll look at this in the next video, but what you can see here is the beginning of what we're going to think about in terms of the process of transcription. We've got the small unit and the large subunit. They're incomplete until this messenger RNA strand comes into position. The messenger RNA strand then is pulled along until it reaches the start codon AUG. This is meant to represent here the transfer RNA, and that transfer RNA is going to carry an amino acid, and we're going to constantly bring in new, this is going to be pulled in, again in the 5' to 3' direction, and we're going to constantly be bringing in new amino acids. This is then going to bind, form a polypeptide bond here as we read the code on the messenger RNA. This is a similar diagram showing you exactly the same thing. I prefer this one. This one is from BioNinja because it shows you that at any given time there are 3 transfer RNA molecules which are associated with the large unit of the ribosome. So this is the E site here, and you can see that this is where the transfer RNA has lost its amino acid. This is exiting. The P site is this middle part here, and the P site is where the peptide bond is going to be formed. The A site, which is this one here, is called the acceptor site, because what we're doing is we're accepting a new transfer RNA into position, bringing with it that amino acid. So what we've got is the E site exiting, the P site where the bond is being formed. As soon as that bond is formed, this will break off. This will then shift across into the E site and leave. That will be pulled further, and then we've got the next A site which is being occupied. And this transfer RNA molecule is being pulled along each time. So I'd like you to stop the video in a moment and look at the McGraw-Hill animation, which shows you this as an animation, and then come back and look at the next video.